Hi, everyone. This is episode 749 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am looking forward to resharing our interview with our guest, Dr. Erica Golemis, originally released on October 16th of 2017. Listeners, Erica is an accomplished cancer researcher. She's studied a variety of topics, including why some cancers are particularly malignant and the genes that cause cancer to change and progress. Her goal is ultimately to identify genes that could be targeted directly with therapeutics to try to reverse this process. And in our interview, Erica shares wonderful insights and stories from her life and research. So get ready for this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of the People Behind the Science podcast. Listeners, today I am excited to introduce you all to Dr. Erica Golemis. So, Erica, thank you so much for joining us on the program today. How are you?
I'm doing fine. Good morning, Marie.
Good morning to you as well. And thank you so much for joining us. We're excited to learn more about you and your work. But I do want to start off by introducing you briefly to our listeners first. So listeners, Erica is a professor, deputy chief science officer, co-leader of the Molecular Therapeutics Program, and director of the High Throughput Facility at the Fox Chase Cancer Center in Philadelphia. In addition, Erica is an adjunct faculty member at the University of Pennsylvania, Drexel University School of Medicine, and the Temple University Lewis Katz School of Medicine. She completed her undergraduate studies in biology and English at Bryn Mawr College and was awarded her PhD in biology from the Massachusetts Institute of Technology. Afterwards, Erica conducted postdoctoral research at the Massachusetts General Hospital Department of Molecular Biology, as well as Harvard Medical School, Department of Genetics, before she joined the Fox Chase Cancer Center. And listeners, Erica is here with us today to talk about her experiences in life and science. So Erica, we're excited to learn more about you and your work. So can you start by telling us what you like to do when you're not doing science?
That's actually a pretty easy question to answer because most of the time I am doing science. I have a lot of commitments at the moment, and so that takes up actually a day or so out of each weekend. But when I'm not doing science, I tend, on the one hand, to read. I read extremely broadly, total bookworm from the time I was able to first read when I was 6. And I like to go to theater, which is a fantastic thing to do in Philadelphia, which is a great city for the Wilma Theater and the Lantern Theater and the Arden Theater and other opportunities. So pretty much strikingly, I suppose that seems a bit weird that I'm living more in the arts and literature rather than in science, but it's a good counterbalance.
No, I love that. We mentioned actually in undergraduate, you majored in both biology and English. So it sounds like that's been something you've been doing throughout your life, is balancing both this arts and science.
Yeah, actually, people tell you that you're supposed to specialize and come to a sense of what you want to do fairly early on so you can focus. And I really had a very hard time doing that as a young person, and I was so indeterminate that I applied both to graduate school in English and in biology.
Oh, wow.
And it was truly what city was the school that had a better reputation, that was doing more interesting thing and that accepted me as a program. And it's like, oh, I guess it's going to be biology.
Almost a coin flip. I like that.
Yeah, it was extremely indeterminate. And I tend to have a fairly cheerful and accepting personality. So I tend to be happy with most things that I land in.
Oh, that is wonderful. Now, you mentioned you're a theater aficionado. Do you have a favorite show?
I'm a complete fan of Tom Stoppard. I think Arcadia is one of the best things I've ever seen. I've had the opportunity to see it twice in recent years, once in New York and once in Philadelphia. It's just absolutely brilliant counterbalancing of the past and the present and academic interests with love and passion and all kinds of things, and beautiful writing.
Very cool. And you mentioned you are a bookworm, and I love getting book recommendations from everyone that we have on our program. So, Erica, what do you recommend that me and our listeners read?
Well, it's very hard. Obviously, if you have many, many things that you like and reasonably eclectic tastes, and different things appeal to you at different periods in your life. So when I was a younger person, for instance, books that I grew up with included basically things that exposed you to other worlds and sort of made you think about the way to live with good role models. Mm-hmm. Mary Renault had a whole series of books about life in classic Greece. Last of the Wine was a fabulous one about growing up in Athens at the time of Socrates, but then heading into decline afterwards. I was a complete science fiction geek in the 70s and 80s, so works of Robert Heinlein, which are a little bit libertarian probably for modern taste, but which had fabulous writing and exciting stories. And The Moon is a Harsh Mistress is great if you're into that. Later into the 80s, writing by Matt Ruff, who is a Philadelphia native. Sewer, Gas, and Electric is a sort of a hilarious social satire that has a lot of scientific elements in it. Neil Stephenson, Neuromancer, basically envisioned the entire internet that we have now before it actually existed. It's astounding to me how much it sort of developed in future, the way he came up with that.
Yeah.
And things closer to science and what I work on, I like basically thinking about science and society. So Neil Postman, who was active at NYU in sociology in the 70s and 80s, he wrote a book called Technopoly, which is sort of an analysis of how technologies basically develop from things that are minor components to having increasing value and prestige, and then if unguarded can sort of take over and sort of rule the way that you're thinking and set priorities that you might not necessarily want to have. You have to balance them with other valuable things in society. And one that's absolutely fabulous is a book by Donella Meadows called Thinking in Systems. I don't know if you've ever heard of that one.
I haven't. I'm going to have to add this to my list.
Thinking in Systems is amazing. This woman was at MIT. She wrote a book called The Limits of Growth, which is a bit more of a bestseller, which I think became very big in the environmental sustainability type movements in the 70s. But this one is really a primer for how to think about how the many parts of complex systems feed into each other. And she starts out talking about, here's an input and here's an output, and then here's a system feed forward and a system feed back. And it starts out being very dull, but then you get past the early chapters and she starts getting into, well, what happens when the system gets more complex, and you start to have sort of sudden threshold effects. And then she takes the whole thing out of an engineering sphere and starts talking about real-world examples as what's going on with the planet. And then she finishes up by saying, well, so we've been talking all about this in sort of hard science-type examples. Let's talk about this with people and political systems and what happens when you have too many inputs and it gets confused and you can't see all the inputs and the outputs. And it starts to sort of lead you to an idea of how some political systems sort of reach the point where you can't impact them in a positive way, or things that you have to do in order to get around hurdles. And it's just really fascinating to read this and then read the newspaper. And of course, I am a sort of news junkie as well, so that also takes up a large amount of my time.
Yeah.
So that's a great deal of fun.
Well, wonderful recommendations. I will add these to our website for our listeners to find there and update my own reading list as well. This is awesome. So it's been great to hear about your life outside of science. But Erica, I do want to chat about your work next. So
okay,
can you tell us how you describe what you do to someone who's not familiar with your field?
So I'm assuming that most people have some familiarity with cancer because it's sort of omnipresent in our lives because we have not yet been successful in curing this pretty awful disease. So for the bulk of my scientific career, which goes back now some 30 years, I've been doing some form of cancer research, although it's sort of varied all over the map. What I've been focusing on for the last several years in my own laboratory has been addressing the issue of what gives some cancers their particularly malignant character. Most people that are listening to this probably know that there's differences in cancer type and differences in cancer outcome depending on whether you catch them early or late. And the reason that the later cancers are bad is that they've gone through changes that make them migrate to other parts of the body where they're not as easily treated. They are resistant to many drugs. They look different. They have different growth properties when you take the tumor out of the body and try to grow the cells. And there's, again, coming back to thinking in systems, there's a lot of evidence that a few changes that occur as the cancer progresses beyond the early stage are causing all of these phenotypes at the same time. So the question is, what are those genes? is there a way, if people are diagnosed with late cancers, to find genes that could be ideally targeted with drugs or targeted directly by some modern therapy that could control them and reverse all the phenotypes, make them be more drug sensitive, remove them, and do this in such a way that you have a therapeutic window so that you're targeting the tumor but not targeting the normal cells of the body and causing terrible cytotoxicity. So we're studying a number of genes. One of them we cloned in our labs maybe 20 years ago now, some other genes also that seem to be involved in promoting the processes of cancer and drug resistance, and one that I'll talk about probably a bit more later that I'm quite interested about that seems to be limiting these processes.
Absolutely. Well, it's wonderful to hear a little bit about your work, and I'm excited to chat about projects as we go through our interview. I think you're absolutely right. I think everyone can understand cancer at least a little bit because so many of our lives have been touched by it. Maybe a relative, a friend, someone we know has gone through this, or the listeners themselves might have experienced it. So
a lot of people that work in this field, actually, they got into it because somebody close to them died of cancer. And, frequently it was quite traumatic when they were young. It's really hard.
Absolutely.
And working in a cancer center environment, I come in every day and I walk through a lobby that is full of patients and their families that are being treated for cancer. And then I go up the elevator and I go into a laboratory that's full of energetic young people that are thinking about abstract science. And so you get a little bit of cognitive dissonance, but I think it's good.
Yeah. Keep you grounded there. Yeah.
It keeps you grounded. Absolutely.
Well, I think that can be motivational too, getting to see the people who your research may ultimately impact their lives. And I love talking about motivational things on our podcast. So Erica, do you have a little dose of motivation you can share with us today, or a quote or a saying that you really like?
That's an interesting question. Typically, I'm interested in too many things. The problem I usually have is maintaining focus. Mm-hmm. And I think that if I were going to answer that, I would simply flip it around and say that I would expect that most healthy people have lots of things they want to do, but they're sort of frustrated by things that hold them back because you have to do administration or you have to deal with really prosaic, boring things. So if I was going to say that, it would probably be "This too shall pass" as a motivational quote, which means basically I may have to spend the day putting together seminar schedules or reviewing a really unexpectedly bad paper for a journal, but it will be done in 2 hours and then I can get back to doing stuff I really want to do.
I like it. I think that's good for all walks of life and all career paths too. So there's always going to be some little bit of drudgery no matter what career path you choose.
I actually think that that's one of the wonderful things about life in science, which is the fact that I probably, if I break down my day, I get to spend a good 50% of the time doing stuff that is really interesting, where I've had creative input that I really want to do. And 50% of the time as a senior person, I don't. I have to do some drudgery. But compared to many other jobs and ways I hear people talking about them, I think that's a pretty good ratio.
Absolutely. So now, can you tell us, Erica, when you were considering career paths in jobs, were there any people that maybe inspired you to pursue science, or any motivational figures that you looked up to as you were growing up?
In terms of people that inspired me very early on, I always liked science, quite frankly, and my parents, on the one hand, inspired me. They both worked in technology, not in science, but they're both engineers. So I always had the idea that working in technology, it was an interesting and a valuable thing to do. A lot of the books that I worked with clearly conveyed that idea as well. Robert Heinlein comes in very well there. In terms of actual flesh and blood human beings, I went to a small school in Manhattan, St. Hilda's and St. Hugh's, which was a very interesting environment. It was the one my parents sent me to because it was low cost and the family didn't have a lot of money at that point. It had a very good reputation, and it was a unique environment because back in the 1960s when I got started, there were, I think, a lot of people that were sort of spiritually inspired or were seeking or whatever. And this was founded as a small teaching order of Anglican hyper-liberal nuns led by a Reverend Mother who had a call. Don't really know as much about her personally, but basically this was a lot of women who had a call to be nuns late in life. Many of them had PhDs, so they were super highly educated. And
interesting,
because it was so tiny, the class sizes were small. So I was being taught by people that graduated from the University of Chicago and other great places, and I didn't know that at the time. But they were really creative. And Sister Dominica, who was the biology teacher, had us doing all kinds of things. She she had us doing dissection. She had us reading things in science, out of science. And in terms of bizarre memories, she tended to be a little bit scatterbrained. And at one point she was looking for the class notes for the day, and she pulled open a drawer and she found her sandwich from lunch, which she had left there quite a while ago. And it had florid growth of all kinds of microorganisms. And instead of going and sort of throwing it out as quickly as possible, she sort of diverted the lesson to be, well, look at this.
Now it's an experiment, yeah.
And so we pulled out the microscope and we were looking at all of the things that were growing on the bread. And I just thought, this is just really, really, really cool. Here's just something that popped up, and we're going to use this and learn about it. And it was fascinating. So that was kind of an inspirational way to deal with the natural world, and sort of took what come to you. And then more recently, and again, more professionally, when I was a graduate student up at MIT, there is a woman who is now a quite distinguished person in science who's actually working in the Philadelphia area, who happened to be in the same laboratory as me and a few years ahead of me. Brilliant. But what was just remarkable to me is that she had every possible thing going wrong with her personal life at the time she was working a few years ahead of me. And it was not her fault. It was just convergence of illness in the family. And she had been working in another lab prior to me, and they ran out of money. So she got laid off right in the middle of her experiments. And her husband had basically run off with somebody else and left her with 2 small children. And she was dealing with all of these things all at the same time. And she made it to work every single day. First thing in the morning, as soon as she had worked out her child care co-op so her kids could be taken care of. And she had at home made up an exact schedule of everything she was going to do. And she was reading the literature in her own time, and she was coming up with brilliant stuff, and she was meticulous, and she was doing important work, and she'd leave on the dot. And she was amazingly effective. She made great discoveries then, and life eventually got better for her, but she had her day scheduled down to, I can now go to the bathroom for 5 minutes.
Oh, geez.
Extraordinary. And I kept touch with her all the years, and she's gone on to do great things. She works at Penn. And she basically said, maybe I was just absolutely uncreative, because when I kept thinking about what I might want to do, I couldn't think of a single thing I wanted to do more than science, so there really wasn't anything else that I had but to sort of buckle down and get through it. She was brought up to expect that life was fundamentally not easy. It was hard, and if you just sort of took that as a given, then you didn't worry about it anymore, and you got on and did what you needed to do.
No time to complain, yeah.
Yeah, so there's another motivational quote for you, which is probably not too inspirational, but it allows you to sort of deal with whatever circumstances you happen to have, and then do what you want to do in spite of it.
Certainly. And now we mentioned you were torn between science and English. What ultimately tipped the scale and got you to commit to science?
Well, once you've committed to graduate school, you sort of are on the path unless something really terribly wrong happens. And I had always liked the natural world. Every animal I ever saw, I liked as a child. When I was in Bryn Mawr, back in the, early 80s, it wasn't a very super strong department, but everybody was clearly interested in problems. We did a lot of observational work back in those days. I spent a couple of summers, on the one hand, working as a research assistant to someone who was doing observational work on a tiny microorganism called a tetrahymena. And it was just beautiful, graphically beautiful, these organisms. And then I spent the following summer doing teaching for postdocs that were in a program that were people that had been liberal arts majors and decided after they graduated that they wanted to be doctors. So they were sort of taking all their science all at once over a one-year course. They were very bright and very interested people, and everyone was really keen on learning a lot of biology. So I was a teaching assistant in a lab for that course, and that was inspiring. So I'm now like 55, and I was just sort of looking back over what has happened in my career, and science has just changed so immeasurably from when I was an undergrad to now. Mm-hmm. And this was completely because of the rise of molecular techniques, the rise of the ability to get sequences. And that was just happening in the 1979, 81. And people began to learn what, on the molecular level, what the sequences were. And that was just incredibly empowering, because first you could see what they were, and then second, you could do something with them in a really targeted way that you couldn't do before. And I ended up at MIT And that was where a lot of that initial work was being done. So everything was sort of groundbreaking. And the person that I worked for was really very inspiring. It's Nancy Hopkins, who is a national figure and worked for women's rights, although that all sort of started after I left the laboratory. We were studying leukemia viruses, so that probably caused some people who listened to have a sort of a sudden thrill of, oh my God, there are leukemia viruses.
Right.
Mostly a lot of what we learned about cancer, and now getting back to ancient history in around the year 1900, so really quite ancient history in science, came from people that noticed that in looking at mice, but also chickens, which are livestock, and other species, that there were clear evidence for viral transmission of cancers. And it turned out that there are quite a lot of these, and people passage them, and then people sequence them all. And so I was doing my graduate work at the time that everybody was sequencing these viruses. And so I spent a lot of time focusing on very small elements of the sequence of these viruses, trying to figure out why some of them caused a red blood cell cancer and some of them caused a white blood cell cancer. And that eventually evolved into a study of, well, what are the actual genes that are causing this virus to act in one cell versus another cell? And then some of those genes got cloned, in part based on my work, a few years after I left that laboratory. And at least one of them turned out to be a major driver of leukemias in humans, just on its work in human cells, getting the virus entirely out of the system. But it was just working in this frontier of stuff that you had no idea what was going to happen, how the experiment was going to come out. And being the only person in the world who really had the answer once you had the answer, that was very exciting.
Yeah.
And after I did my graduate work, I really did a major change because I had spent a lot of time studying viruses and coming up with some insights into that. And I wanted to try something really, really different where I would get closer to the engineering front. And instead of studying what the system was trying to tell me, I was going to try to see if I could use all of this technology that was being developed and turn it into a useful tool that would allow us to learn more. Because sitting there, you could see what the invention of something useful like sequencing had done, just revolutionizing the whole field of biology, which has been ongoing. And I went right across the river, one stop on the Boston T. So instead of getting off at Kendall Square, I was getting off at Charles Street and went over to Mass General and worked with a young faculty member there named Roger Brent, who was absolutely brilliant, and spent the next 5 years of my life working on development of a system called the yeast-2 hybrid system. And there were multiple forms of yeast-2 hybrid system. Other people were developing these principles as well. But the idea was we were building a system that was sort of using yeast, the type of yeast that bakers use or beer makers use, using it and engineering components in it so it would be sort of a test tube that would allow you to study how specific proteins interacted with other specific proteins. And so it's a little bit hard to explain this without being able to draw a picture that people could be seeing at the same time. But basically, it was reasonably cheap, and it allowed you in some ways that you use the system once we got it to work. So we ultimately did get it to work, although it took 4 years. It allowed you to screen through millions of different clones of different human proteins to figure out who was talking to who. So it became this real enabling technology that allowed you to say, okay, I've got my gene, but instead of just studying my gene and the protein it encodes in isolation, now I can take that protein and I can find all of its partners. And by doing this and combining notes with the community and with everything, I can put together a really dense web and figure out who is talking to who and how signals are propagating through entire organisms. And that was extremely exciting. So I did that for a long time, too. And at this point, I'm totally hooked on science, and then went on and developed things in my own laboratory afterwards.
So now, speaking of things going on in your laboratory, Erica, do you have a favorite project that you're working on right now that you want to tell us more about?
Well, it's very dangerous to say that you have a favorite project because I have 4 students that are working on 4 different projects.
I know.
And that's just asking for trouble.
True. Do you have a project you'd like to highlight today?
I'll highlight one, which is a theme that we've been working on for several years now in various elements of it. So everything that I work on has a little bit of a history. So let me give you a little bit of background to why we got interested in this. Mm-hmm. We've been studying, as I said, a bunch of oncogenes, so cancer-promoting genes forever. And one of them that we started studying is something called aurora kinase. All the genes have these really bizarre names that come about through whimsy and historical reasons and so forth. So
yeah, they're kind of fun. I like them.
Yes. And this one's called aurora kinase because basically if you mutate it and look at what happens to the cells as they go through mitosis, all of the DNA is sort of splayed out in a really bizarre way. And it looks like aurora borealis. So that struck someone poetically when they were naming the gene.
Gotcha.
But that aside, so I was studying this gene, which a lot of people were studying in the context of a cancer target, because if you make too much of it, which happens many times in cancer, the tumors basically get genomic instability and aneuploidy. So we were studying the function of this gene and reading very broadly in the scientific literature. And I'm sitting in a community of people where people are taking drugs to clinical trial to inhibit this because everyone thinks it's blocking cell cycle and mitosis. And so we found this provocative paper where someone was studying green algae, which sort of emphasizes the value of studying model organisms. And they found out that it was working in a totally different way. Mm-hmm. Which is through not being a cell cycle regulated gene, but being involved in response to certain classes of environmental stimuli. And then when it's induced, it caused loss of a structure on the surface of the cell called the flagella. So we started studying this process to see if it was relevant to human tumors and human normal cells, and in fact it was. And this took us into the biology of a whole second function that was sort of underappreciated of this gene in removing the cilia from cells. And cilia are really, really interesting because the cilia is a point where a lot of the signaling system of the cells sit. And if you want to visualize it, the way to think about it is an old-fashioned antenna sticking off a TV set. Really, it's just like a little spike that's sticking out outside of the cell. And the cell uses this to display things that grab signals coming from the environment, and that allows it to change its behavior in really interesting ways. So we've been studying this for a while. And the cilia is interesting for people in cancer because of what it's doing to signaling in cells, but it's also really, really interesting to a large community of people that study things called ciliopathies because there are diseases that target the cilia specifically, and one is something called polycystic kidney disease. And that is one of the most common inherited syndromes that people work on. It affects about 1 in 500 people.
Oh, wow.
It's mutations in a gene that cause proteins that are linked intimately to the cilia. And if you have this disease, which is a dominantly inherited disease, so basically if you have it, your kids have 50% chance of having it as well. It basically causes your kidney to fill up with cysts and lose function in middle age. And so these people end up on dialysis, or they end up having to have a kidney transplant, and they have other health problems as well. So it's quite a serious disease.
Yeah.
So we started being fascinated by the fact that there were a lot of common things going on in the signaling at the cilia that were related in cancer versus in polycystic kidney disease. And that was really surprising to us because the way these diseases manifest in people is quite different. In the one case, you have a kidney that fills up with little bubbles and pockets. And on the other case, you have a tumor that the cells basically lose all their identity and they metastasize. But if you look at what's going on at the molecular level, there's a lot of commonalities.
Gotcha, yeah.
So what really, really captured my imagination, we've been studying back and forth what's going on with the signaling, but there was this fascinating paper a few years ago where people that were epidemiologists at the National Cancer Institute got together with people at one of the major PKD centers that are out in Kansas. They're just wonderful people. And they did an epidemiological study of people that had PKD and then looked to see whether they had greater or less risk of cancer. Mm-hmm. Large studies, so like 10,000 people with PKD and 100,000 people that were relatively matched to them who had had cancer. What they found was this provocative hint that if you had a gene that put you at risk at PKD, you had a reduced risk of colorectal cancer, which was fascinating.
Yeah.
Why would that be? And so we've been studying that, actually. So this is work that I don't yet have funded. This is work that I have not yet published on, but where we're getting some really provocative things in the laboratory, because basically we've been doing preliminary experiments with one of my colleagues here who was an expert in colorectal cancer mechanisms. And we've been crossing mice that are predisposed to polycystic kidney disease to mice that are predisposed to colorectal cancer. And we have some early data that is indeed suggesting that having the one disease or being at risk for the one disease is actually reducing a number of things that are going along with cancer risk. And so we don't understand how this is working yet, although we have some ideas. But it's quite exciting because if you can understand how this genetic defect is causing a possibly beneficial effect on the other end, this might suggest ways that you could help with cancer prevention somewhere along the way.
Absolutely. Well, Erica, this is a phenomenal and exciting project that you described here, but I know not every day can be full of exciting discoveries. We mentioned there's some mundane tasks you have to do as a scientist, some frustrations that you encounter, and some challenges along the way. And I do love to talk about those on our program as well, so our listeners get the holistic picture of life as a scientist. So are there any major failures or challenges or problems that you faced in your career, maybe ones that made you think back to that wonderful potential career in English?
So this is an interesting thing about life and science, which I actually talked about to my son and some of his young friends. Mm-hmm. I mean, you fail all the time. You fail on small levels and you fail on large levels. So this is something where you basically have to separate your ego entirely from failure, or you could not do this job, which is a little hard to do initially, but ultimately it's somewhat liberating. Mm-hmm. What I mean by that is that you fail every day on levels from the I had an idea, I tested it, I was wrong, right, to the I had an idea, I spent 2 days setting up the experiment, and one of the reagents that I'm using, something has gone wrong with it, and it just plain technically failed for totally mundane reasons, and I'm going to have to spend the next 3 days trying to figure out how to troubleshoot this so that I can actually ask the question that I wanted to ask. So that's one level of failure that you have to put up with all the time, and the compensation, of course, is that when you get it to work and you proved that something clever you came up with and insightful you came up with was right, well, that sort of makes up for it. Right? Sure. But you have to deal with huge amounts of failure all the time, and sometimes it does in fact build up. So one memorable experience to me that I turned it into something productive was I was back in graduate school. My main project was studying how these leukemia-causing viruses cause red cell versus white cell leukemia, and one way that I was doing this was by making some fairly complicated DNA constructs that would allow me to make viruses, that would allow me to move pieces from one to the other and back to the other, and it was very finicky work. And because of the nature of the sequences I was working with, there were 2 or 3 clones that I needed to get to finish my thesis because they were sort of testing the key point. And it just failed. I tried to make these things for 4 months, maybe more, and things that should be basic just failed to work completely. Oh, wow. So it's very hard to just set it up again. But, I was doing that. And so the way that I sort of started dealing with it at that point is saying, okay, I have to set this up. I'm going to have big gaps of time in this procedure because you set up a reaction and then there's a 2-hour gap or something.
Right, you have to wait, yeah.
So I'm going to do something with this time other than just hate my life, which is a tendency at that point. And so I had mentioned that sequence information was really taking off like crazy at that point. So I would go to the library and I just decided I have to write my thesis. Hopefully this is going to get done and I can write my thesis. So I'm going to educate myself about all of the retroviruses, which was doable because there were, I will tell you, 37 of them at that point in life.
Really? Wow.
Because basically we go to the library. It's so easy to find things on the internet now. Back in the 80s, no internet, no real organized catalog.
I wonder if many students have been to the library now, right?
Oh my God. And there would be books that would be bound as back issues and they would weigh like 500 pounds. Mm-hmm. So you'd go and you'd sit there and you'd be searching through the abstracts of what papers had been published, and you'd be looking for keywords, and then you'd go to the stacks and you'd pull back these heavy books. And so eventually I pulled every known sequence of a retrovirus, and then I'd go back and have my reaction fail again. But I started looking at them all and started writing up notes, and then I realized that basically I had been sequencing so much, I had memorized large parts of the gene sequence.
That's impressive.
And I realized that I'm looking at virus sequences from chickens and from cats and from other animals that were fairly distantly related that people had been sequencing retroviruses from. And I realized that I recognized parts of it by eye because I had been doing it so much. And then I thought, could it be that the better way to do this experiment is not just to cut and paste, but to just go and line up these sequences and see if I can actually make some sense out of it. So people do a discipline called bioinformatics now where they use high-powered computational tools and they use big data and they devise things out of it. So this basically, the word bioinformatics did not yet exist, but what I ended up doing was getting lots of graph paper, pasting it together in a long sequence and by pencil, lining up all of these sequences by hand.
That is amazing.
And it was 15 feet long and it filled my entire living room at my house, and my husband gave me a hard time about it and my cats kept running across it.
I hope you kept this, Erica. This sounds amazing.
It's around here someplace. I know it's in my office, but my office is trashed. So, but that turned out to be the piece of work that got most cited out of my thesis when I finally convinced my advisor that it was worth actually publishing something that was just sequencing, because basically when you did this, there was a core element to all of the enhancers, right? And it was matching what I ultimately, after much pain, managed to make these viruses. The same sequences were important. And this was a guide that allowed one of my colleagues, actually the colleague who I was extremely impressed by and was talking about a few minutes ago, to go out. This helped her figure out what were the key elements to go out and then find the protein that was the leukemia-causing gene. So it worked out, and it was just born out of complete and total frustration.
Yeah.
So you just do something different.
Well, you handled that frustration in a very productive way. I think most of us would have just sat there hating our lives for the 2 hours. And
this is what you have to do. You just do something different, because you can't fail at everything all the time if you keep starting new ideas. Certainly a high rate of them are not going to go anywhere. You're going to run out of money. Mm-hmm. But things that are based on reading and thinking and going back to the literature and being creative are much more likely to yield something productive at the end of the day.
Well, I'm so happy to hear this story has a happy ending indeed, and you were able to get something out of this frustrating experience. And we do love celebrating these successes in science in all of its many forms. So do you have another success story, Erica, that you want to share with us today about your experiences?
So, again, I will talk about stuff that I think is a success, but it's not published yet, although I'm hoping it's going to be soon.
Sure.
The country, the science establishment, the NCI, everyone has decided to make a massive investment in doing big science, right? Mm-hmm. And big science is something that I have a sort of a love and hate relationship with, because on the one hand, a lot of times you just churn enormous amounts of data and you don't really do something that is beneficial with it. You just develop huge resources, and then sometimes people don't really find a home for those resources.
Right.
But a lot of times, if you can figure out how to go in there and look for specific test hypotheses, then you can come up with things that you would not be able to possibly generate on your own, because you can't possibly pull enough money into your laboratory as an individual working in a small institution. So my advice would be to find the bioinformaticians and biostatisticians that work near you and become friendly with them. Mm-hmm. So one thing that I think is successful lately is the nature of my job has me sort of sitting at the hub. I work between my group and some others that do wet laboratory work and work with cells and mice models and things like that. But we also work with people that are involved in structural biology, people that are involved in sequence staring, and a lot of medical oncologists that are interested in understanding the specific defects in tumors of the patients that come in with the idea of finding therapies, and figure out which patients are going to respond to which therapies and who's got a good prognosis and who's got a bad prognosis. So these are all issues that we all care about a great deal at the center. So I've been working for a number of years with people that work at neck cancer, and we have some bioinformaticians that we work with on that. And I've been involved in a project that I think we're going to get published soon, where one of the bioinformaticians basically identified a new way of analyzing large data sets that involves integration of how genes are expressed, copy number variation, DNA methylation. So this is getting a little bit technical, but all kinds of different really large data sets that are out there and are publicly available. And so he decided to apply this in the context of head and neck cancer. So by basically crunching a large amount of data, this came up with a suggestion of a very specific mutation, which I shouldn't talk about yet because I don't want people running out eating themselves sequenced or something.
Sure. Don't let the cat out of the bag early. Right.
Yeah, and I wanted to make it finally through peer review, which it should do in the next month or so. Mm-hmm. But basically, he identified a gene which, when mutated, had a very strong effect on prognosis and therapeutic response in one specific type of head and neck cancer. So that was quite exciting. And then because when you're doing this, you do this really rigorously, you have to find a different subset of patients because it could possibly be a statistical artifact if you just find one set online. And so that sort of led us to do what we always do to get lots of data. We collaborated with people at this institution, at Johns Hopkins, and we pulled up a really large data set that was completely independent, and we went in with the gene signature, and we found the same result.
Oh, wow.
So we're very excited about that. So we know the nature of the gene, and this is now sparking collaborations to figure out if we can exploit this in some way to figure out which patients should get which cancers, and it suggests a lot of mechanistic experiments we might want to do with colleagues here in the laboratory. So that's sort of pulling all the pieces together and getting sort of teams to work together.
Well, congratulations on this exciting success. So what was going through your mind, Erica, when you saw the results validated in that second set of data?
There's no feeling like it.
Yeah.
Not only did it mean that you haven't just wasted a year and a half of your time, which is always a possibility.
And quite a lot of money, right?
Well, time, money, whatever. But it also means, yes, this is real and this is really important. And we are the only people in the world who have figured it out. And you feel like you've actually done something that is productive and exciting and opens new doors. And we get that kind of a sort of huge feeling of I haven't wasted my life every now and then, which is quite exciting.
Right. Well, wonderful to hear about this exciting success, and our listeners and myself are eagerly awaiting the paper now. Hopefully we'll get that through soon. Mm-hmm. And we've talked about some different aspects of your career, Erica, but one of the things that I love chatting with scientists about are some of the opportunities that they get to travel, to work with different people, to see different places. So do you have a favorite place that your science has taken you?
One of the more interesting places that it has taken me has been to Moscow and Russia several times. And the reason that it has done that is that back in the 1990s, we were trying to strike up collaborations at our institute that would lead us to be a more prominent destination for talented graduate students and postdocs. And we were getting a lot of people already coming out of the former Soviet Union as it had been breaking up. Mm-hmm. So we reached back and formed partnerships with a number of laboratories, initially in Moscow. Now we have collaborations in other cities around that have very, very strong traditions of scientific education and are interested in building bridges and having people that come and train in the U.S. Some of them go back to Russia, some of them stay in the U.S., some of them go to Europe. But basically, this was a chance to go off to another city and learn how people do science in a different environment and basically see just a little bit of the culture. I mean, you can't learn a lot in 6 visits, but it was quite exciting.
Sure. So what was one of the most striking things about Moscow for you?
On the one hand, so alien, and on the other hand, so very familiar, because I grew up in New York City, and a lot of the ways that the people are remind me, I think there must be a lot of Russian immigrants or something to New York City. Mm-hmm. So parts of it seemed very familiar, but then parts of it were absolutely remarkable. We did a tiny bit of sightseeing. We went to St. Petersburg. We were there for one visit. We saw the Hermitage and Peterhof, and you go walk through these spectacular palaces, and you think, I understand why there was a Russian Revolution, because it's such an appalling concentration of wealth that you can imagine. But on the other hand, all the scientists that we met were fantastic. And I mean, aside from the hard work, everyone also was extremely cultured at the time in science, because I think you basically had this sort of academic class, and everyone knew the complete Russian literature, and they knew poetry. And if you walked around the streets, there were monuments to the famous authors, and that was really interesting too. And it was clearly a lot of affinities that were going on, and a desire, since this was in the immediate post-Cold War, to try to figure out how people could work together to invent something new, to get out of the stasis that the society had been locked in a sort of state of decay for the previous couple of decades.
Yeah, how do you move forward? Yeah.
Mm-hmm.
Well, very cool. Great to hear about this travel experience to Moscow, a place that I'm sure many of our listeners haven't had a chance to go. So it's great to get this little window into your experiences there. But I also love chatting about some of the people that you get to work with as a scientist. And you hinted that these people you met in Moscow were phenomenal and it was wonderful to work with them. And I love showing this human side of science and showing science as more of a social endeavor, because I think not many people appreciate that.
Oh, it absolutely is.
It absolutely is. So do you have a story, Erica, maybe a quirky tradition or a funny memory or just something fun that you've done with your colleagues?
This lab is fairly hardworking, so we don't go out and play a huge amount. Mm-hmm. Certainly, back when I was a postdoc and things were a little bit less frenetic than they've gotten more recently, we used to have lab outings all the time. We would go out and we would play mini golf. We would out and we would go hike. Science used to have a great deal of low comedy because back in the 90s and so up until the workplace got more separated from after hours, people would always go out to beer hour on Friday night, listen to a seminar, come back into the laboratory and run their gels backward or do stupid things with their experiments. Mm-hmm. We don't do that so much now as everyone has gotten more sober in the workplace. I mean, in terms of quirky things that we do in this laboratory, we are mostly professional. I mean, I suppose one of the more bizarre things that we do is we always celebrate a holiday lunch as a group, but where most people would normally put that December to January, we always have our holiday lunch in February.
And is it like a Valentine's lunch? Or
Well, it's theoretically an end-of-year celebration lunch, but the reason we do it is my laboratory has people from China, it has had people from India, it has, in fact, a lot of people from Russia, and everyone's traveling. So the travel season is, since everyone's on a different schedule with their different cultures, people start going out in early December, and some of them are going out in January. And what with coming and going, we're usually not here in the holidays as a group.
Right.
So February is just a terrible month anyway. The weather's lousy, so we always have holiday lunch around February.
Yeah, you need a bolstering. Yeah, right. So what does this holiday lunch usually consist of? Do you go out to a nearby restaurant or?
Sometimes we'll go out to a restaurant. Sometimes we will just have people do potluck. Sometimes we will go and we'll hit up the Russian deli, and we have lots of pierogies and things from the country of Georgia that they specialize in our area. So, yeah, it varies from year to year and depending on the preference of the people that are in the laboratory.
Well, this sounds like an excellent tradition, Erica. I love it. And we've chatted about some of the amazing work that you're doing in your laboratory now and the wonderful people you get to do it with. But I love challenging the guests on our program to think big with us, to dream big. So if we gave you everything, all of the funding, staff, technology, took away feasibility constraints, is there one question, Erica, that you are just dying to know the answer to?
So most of the effort that we have and most of the money that we have is focused on dealing with cancer after it has developed and frequently after it's quite late. And there's a large school of thought that's out there which basically says that so many changes have occurred at that point. It may be much better to actually focus on understanding what's happening very early when cancers are forming and to basically work on the prevention end of things. And that is insanely hard to do because there's so much variation in the population, and people are not mice. There's a quote for your audience. People are not mice. Right. You can't make them do things. And everyone has a different life experience, and everyone has a different biology. I've been working, actually, after many years away, I've reconnected with my thesis advisor because she's really excited about a project that she's doing, which is trying to figure out what percentage of cancers are preventable. And she's working with epidemiologists and exploring roots of things that might be possible contributors to cancer to try to get an actual hard estimate of what are things that we might do before cancer is caused that might help reduce their incidence. And I'm interested personally in the mechanistic end of that. And so in terms of picking apart, this would have to be a huge study. It's probably not doable, but one theme that's really been emerging in cancer has been the role of inflammation in cancer incidence and in treatment and in every aspect of cancer. The fact that if you're in an inflamed environment and your immune system is not working properly, this does things to how cancers grow and how they respond to therapy. So head and neck cancer, which is one, once again, that I worked with a bunch of great medical oncologists, that's a cancer that is clearly got environmental factors or behavioral factors that lead into it. Smoking and drinking are major drivers of head and neck cancer. In younger patients, human papillomavirus infection is a major increasing stimulus. So these are things that are related to behaviors, but they all also are related to the idea of inflammation. And so If you think about the environment, there's evidence of things that we dose ourselves with and that people in the population dose ourselves with that promote or block inflammation. So like people take aspirin because it's good for colorectal cancer, but it has some risks because it can cause erosion of the gut, but it reduces inflammation. So if you sort of extend this and you think about, well, how are we modulating our inflammation? People take allergy drugs. My voice probably sounds a bit scratchy right now because I have horrendous allergies at the moment because I live in a part of the world with goldenrod. People live in areas that have more allergens or less allergens. People are dosing themselves with quite potent compounds like Allegra or Singulair or Benadryl. People have acid reflux because they eat too much or because they're just unfortunate, and that's another thing that causes inflammation. Red wine, it has compounds like resveratrol in it, which are good for you in long life, but it also can be pro-inflammatory. So all of these different things that are the diverse factors that are going into the population are clearly in subtle ways modulating the inflammation in different parts of their body. And how is that impacting cancer? It's hard to know because for the most part, people don't record, are you allergic? What over-the-counter medications are you taking? All of this kind of stuff when they're in a cancer treatment or when they're in a cancer prevention program, because it would just be a questionnaire that's 20 pages long, right, or 50 pages long. But it's fascinating, and every now and then you get a really tantalizing result coming up. Like there was just a paper out in the New England Journal where people were studying this new anti-inflammatory drug called canakinumab, which all the new drugs have terrible names, so don't have to remember that.
Right. They sound like a disease themselves, yeah.
Yeah, but they were studying it for heart disease because of a specific role of this thing that it targeted in heart disease, and they unexpectedly found that there's provocative evidence that it's reducing risk of lung cancer. And that's really one of the kinds of things that makes you think that there could be things that are going on. So these would be large studies, but...
Yeah, well, this is a wonderful question. I think even the fact that we can talk somewhat realistically about the possibility of cancer prevention is just amazing in this day and age.
Yeah, and it's frustrating because, in a sense, there's been a poisoning of the well because we keep having studies where the New York Times reports that somebody said, eat this and your risk of X will go down, and eat that and your risk of X will go up, right? And so people are whipsawed as what should they do with diet, what should they do with other lifestyle things. And these are small studies when they're confounded frequently because they don't have statistical power. All of these, there's probably something real going on, but it's going to be scientifically almost impossible to come out with a definitive answer anytime soon, which is probably one way in which big data is going to be useful in another 10 years as we keep recording things.
Absolutely. Well, it's been wonderful to chat about the exciting possibilities in the directions that this field is going. And I do love to give our listeners, though, advice during our program that they can apply to their own scientific journeys. So, Erica, is there one piece of advice that you received at some point in your career that you want to pass on to our listeners today?
Sure. One of the things that actually just went by in passing at the time, but for some reason it stuck with me, is about, 15 years or so ago, I was trying to do something, starting a program here, and I was just going out of my mind, running around, talking to senior people since I was pretty junior at that point, and I ended up with the chief finance officer at the place, the budget administrator, who was a very wise man, Bob Spallone, who is now retired, and he basically sort of sat me down in his office and said, it's easier to get forgiveness than to get permission. And that's basically true, which is that if you have something and you are really, really sure that it's going to work and you're just a little bit worried that maybe somebody's going to get upset about it, that sometimes you should just go ahead and do it and prove that it actually did work, and then say after the fact, well, look, we started this great thing and aren't you glad I did it now? As opposed to trying to convince someone without hard data that it actually was a good idea.
Well, I love that advice, Erica. Is there any other last piece of advice you want to give our listeners, or a last note of inspiration you'd like to end our conversation on today?
I think it's a bit of frustration because I read about STEM frequently, and I read the Chronicle of Higher Ed, and I read in the news, and I listen to people talking about STEM jobs and science jobs. And what frustrates me about all of that discussion is that it doesn't really seem to be talking about it properly, because the way we frame the discussion is, it's a great job. you're going to have good career prospects. It's going to be secure. And it seems a little bit boring if you're a young and idealistic person. And another thing is that people get the idea that it's very hard to do science because basically they run into the vocabulary. I'm sure I've dropped a few words in here that are not completely familiar, but because you're working in a subdiscipline, you get a very specialized vocabulary, and that is off-putting to people. So they feel somewhat alienated from what people are doing. But the actual concepts are frequently quite simple behind the vocabulary. So what you really want is to just encourage people to have sort of an adventurous spirit and to have a creative spirit. And science is a desire to discover. It's part hard work. It's part tolerance of failure. But then it also has elements like you have to read all the time. You have to write all the time. Everyone in my lab works on writing reviews when they come in just because that sort of gets them trained on reading the literature and then writing intelligently and summarizing things. You have to be able to, at some level, master public speaking, even if you're an introvert, which many of us are.
Right. I'm guilty myself, yeah.
So it's not the career that is portrayed to young people, which is really a pity. And I think that a lot of people would love doing this, and it would perhaps be a slightly different set of people that would be turned on by the creativity.
Well, Erica, I am so glad you brought up some of these points because I think this is a lot of the goal of our podcast is to show people this other side of science or to give people this more holistic view of the career of a scientist. So listeners, I swear I didn't pay her to say all that. So that's phenomenal. Wonderful note for us to end our conversation on, Erica. Can you tell our listeners if they want to learn more about you and your work, where should they go?
Well, I tend mostly to have my life organized around my email, so they could write to me at my work email. It might take me a little while to get back to them, but I certainly would answer if someone wrote in. That could probably be found by finding my laboratory on the Fox Chase Cancer Center website. So it's my first name, Erica, spelled with a C, period, Golemis, Golemis, at FCCC for Fox Chase Cancer Center.edu.
Perfect. Well, listeners, definitely read up more on all the amazing projects that Erica is working on and get in touch if you have questions. And Erica, thank you so much for joining us on the program today.
Well, thank you. And it's been really a pleasure talking to you and having a chance to talk about our work.
Excellent. Well, it has been wonderful to hear about your exciting life and the exciting work that you're doing. And great to have our listeners here with us today as well. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.